Hi there, Ms. White here. Welcome to part 2 of the regulation of the cell cycle and what can go wrong. This is what we started in class on Tuesday, and we're going to continue it now. So make sure that you have your lecture packet handy, something to write with or highlight with as we go through the rest of the lecture packet. Okay, so period 2 has not seen this slide yet. Period 1, I know we already went through some of this, but I'm going to go through it again. So first of all, let's go ahead and just read through this first section and get your highlighter handy and highlight what you think is important. But remember that we went over the checkpoints. There are these points at which a cell has to make a decision about going forward through the cell cycle. So what determines if that cell is going to do that? What basically determines if it's okay to pass a checkpoint, right? And so these questions are answered by intracellular, meaning signals and conditions inside the cell, and extracellular, which would be outside the cell, conditions and signals. One of those signals is called a growth factor. It's important to know that growth factors are a protein that would be released by specialized cells and trigger cell division. There's also going to be surface proteins on the cell membrane of the cell that tell the cells to stop dividing if the environment gets too crowded or if they're touching each other, indicating there's not enough room. There's going to be another category of proteins, again, enzymes are proteins, called kinases that are going to provide the energy through phosphorylation for many of the processes that happen. We know that dividing is going to require a lot of energy and resources. So first of all, let's just review some vocab here. Phosphorylation. So think about it. What does that mean? Now you can always pause the video if you ever want to stop and think or write down some additional notes on your lecture packet, but hopefully you're thinking about what phosphorylation means and remembering that it means to add a phosphate group to a molecule, which is then going to increase the energy of the molecule. The other key point from this initial reading is that we mentioned a lot of different proteins. So remember, maybe add a note for yourself that all of these proteins that we've been talking about are coded for by genes. And again, if those genes get mutated, it could affect the function of that protein, which is going to affect this whole cell cycle process. Okay, so now let's go ahead and take a look at this model. So we've seen here we have some vocabulary, some names, cyclin and kinase, and it's showing us a graph of the activity of those molecules. So first thing you want to notice is, sorry, this thing keeps popping up, is what are these different molecules representing down here. So we see the first one is, you can't see it, that thing's in the way, but it's called cyclin-dependent kinase. That's going to be that dotted line right here. Notice what it does throughout the cell cycle. Stays the same, right? And so down here we're seeing, okay, during G1, S phase, and G2, it's all going to stay constant and even through mitosis. So this cyclin-dependent kinase molecule, CDK, stays constant. That's important. The next one is the solid line. It is a molecule called cyclin. Cyclin increases throughout the initial part of the cell cycle, G1, S, and G2. And then all of a sudden, it gets to this point, it reaches a threshold where it seems like something's happening, and all of a sudden, it's going to decrease at the beginning of mitosis. Then it starts again, so we get this cyclical effect of cyclin. That's where the name comes from. It increases during interphase and then decreases dramatically. And then the last thing we see is this thing where all two of those, the CDK, which is the clear box, and the cyclin combine along with a P. Remember, that's a phosphate group. When those combine, it is known as a maturation promoting factor, MPF, and that is this part of the graph here. And notice again that the pattern kind of mimics the movement of the cyclin. So as cyclin increases, obviously because cyclin is a part of MPF, then MPF is going to increase, and then when cyclin decreases, the MPF has to decrease also. So that's what we want to notice about this model right here. Okay, so you have questions 11 and 12 on your paper. We just extensively talked about that model. So pause the video right now, go ahead and answer and write down some information for 11 and 12. Okay, welcome back. Now you can look at questions 13, 14, and 15. And again, we just talked about this diagram. You should be able to answer what's going on with the concentration of the CDK and the concentration of the cyclin, and then why MPF is going to also have that pattern. So pause the video and write down your answers for 13, 14, 15. While you're taking a break from the video, I also want you to go to your Canvas page, and on the resource page that says Control of the Cell Cycle, there's a link to this animation, and I want you to pause the video and go watch that so that we can talk about what you saw in the animation. Okay, welcome back. So here, hopefully you just watched that animation, and here are a few screenshots from that animation that I wanted to emphasize. So notice here that this blue part of the diagram is supposed to be the CDK, the cyclin-dependent kinase. And here at the, towards the end of interphase, it's binding to the cyclin, becoming the mitosis, or also known as maturation, promoting factor, the MPF. Okay, so we're seeing that kind of mimics this part. Here, let me get a highlight. Okay, so that kind of mimics this part of the graph where these are both increasing because we have a lot of the MPF, okay? And then part 2 from the diagram shows, okay, that triggers the cell to move into mitosis. So right here, there's enough of the MPF, enough of those active molecules that can then provide energy to get things moving, so it makes the cell move into mitosis. But the other important part of that was that this also activates another enzyme protein called cyclin degrading protein. So degrading means that it's going to break down, okay? It's going to break down the cyclin. So next we see that cyclin starts to go away, and that's why we see that drop right away. So it kind of mimics, the pattern that we always talk about for homeostasis. If there's an increase, then there's got to be a decrease. If there's a process that's starting, we need a way to turn it off. And so this is how the cell can chemically be told to move from one stage to the next and to finish up mitosis before going into the next part of the cell cycle, etc. Okay, so here's question 16, and considering model 1 and 2, which checkpoint in the cell cycle is regulated by concentration of MPF. Think about that, thinking about where in the diagram things are spiking, and write down your answer for that. I'm actually going to have to continue this part of the lecture in the next video, so check back for the next video posted as a continuation for this lecture.